Hi everyone, welcome to the first scientific seminar for the year. I'm Caroline Miller. I'm here to welcome our guest, Dr Anne-Lise Chaber. So firstly, I'd like to acknowledge the country we're on. So we're on the land of the Kaurna people, who are the traditional custodians of the Adelaide region. I'd like to recognise the Kaurna people's cultural, spiritual and physical and emotional connection with the land. I'd like to honour and pay my respects to elders past and present and all generations of Kaurna people now and into the future. And I'd like to make specific acknowledgement of any Aboriginal and Torres Strait Islander people who are in the room or who are listening online. So it's my very great pleasure to introduce Dr Anne-Lise Chaber. So Anne-Lise is a One Health expert. She's got a joint appointment at the School of Public Health, which is the school that I'm in at the University of Adelaide, and the School of Animal and Veterinary Science. So Anne-Lise's research is focused on preventing, detecting and managing infectious diseases at the animal and human interface, which I'm sure we can all appreciate is highly topical at the moment. And her ultimate goal is to safeguard public health and biodiversity while managing the risk of zoonotic diseases and pandemics. So today Anne-Lise is going to be talking to us about the scientific adventure of the COVID sniffer dogs, infectious disease screening using volatile organic compounds, a paradigm shift. So please join me in welcoming Anne-Lise.
Well, thank you very much for the introduction and thank you very much for having me here. It's actually a pleasure to speak about this project, and I would like to take you on the journey of and this adventure of the COVID sniffer dogs. By training, I'm a veterinarian. That was many moons ago, and then I did a PhD in epidemiology, and as Kate said, I'm working on disease at the wildlife-livestock human interface. So I'm not a dog specialist, but I became much better through the life of this project. So first, why do we need to detect disease early? Well, we need to detect disease early, first for the benefit of the individuals, so they can have access to the best treatment in a timely manner. But it's also important at a population level, so they don't infect other people. And post-detection isolation is very important to flatten the epidemic curve. I'm sure I don't teach anything there. So to do so, we need very fast and reliable screening tool. And I got that really clear in my head when I was working in the UAE. I worked in the UAE for 8 years, and I was working on the MERS coronavirus outbreak in Al Ain, which was the epicenter of the outbreak of the MERS coronavirus outbreak in the UAE. At that stage, we were trying to understand which animal was the source of the infection. So we were sampling bats, we were sampling camels, and even so I didn't have much to do with the human population. We were looking at the infection rate. MERS coronavirus was killing 1 person out of 3. So the reproductive ratio was a bit less than SARS-CoV-2, but the fatality rate was much higher. So the problem of the screening tool was very important then. For MERS, like for SARS, we are relying on different conventional disease detection methods. So first one is a PCR, or polymerase chain reaction. So we all know it's a molecular test that is looking for genetic material. And we can also use antigenic tests that can detect the presence of specific proteins of the virus. The problem is that PCR is a reliable tool, but it's not fast enough during a pandemic and during an outbreak. Antigenic tests or RAT tests are quite rapid. The reason why we are calling them rapid antigenic tests, but they're not always very reliable. So we were left with this dilemma of not having the tool we needed to actually act quickly enough in an outbreak situation. So I scratched my head and I was like, we need to find another tool that is going to be both fast and reliable. And that was, I think, in January 2020, the very beginning of the pandemic. I was like, what tool are we going to use? I looked at literature and I started to scramble ideas from very far and from very long time ago. I even looked at what Hippocrates, a Greek physician, what we call the father of medicine, was advocating. And it actually came as a surprise that 400 years before Christ, he actually taught his students how to sniff their patient breath for the aroma of disease. His students were encouraged to smell the sputa, blood clots, ear discharge, urine, excrement, and vomit to aid diagnosis of disease. Medical practitioners have put this tool in the back burner for a few decades, or actually a few thousand years, and I can understand why. But more recently, we've been using animal to detect disease. We've got some anecdotal evidence of dog detecting cancer. The first one was actually a publication from 1989, where a dog kept poking the back of his owner. So the dog, there was a cloth, the owner had a jacket, and the dog kept going to the same point at the back of her back. So actually, that prompted her to seek medical advice. And upon seeking medical advice, she was diagnosed with a melanoma. This dog has never been trained to detect cancer, but his sense of smell was, or he used his smell to actually indicate that there was something that was abnormal. Much later, in 2013, we had another case of someone who actually had a lesion behind the ear. Another dog kept actually going and licking the lesion behind the ear, which prompted again this person to actually seek medical advice, and again it was a melanoma. So there is something that dog can sense when there is a disease happening. We've got now a lot of evidence for many diseases that actually dogs are very good at detecting some types of cancer, including prostate cancer, all the skin cancer. A lot of studies are looking at ovarian cancer, and now dogs are being actually trained to detect neurodegenerative disease such as Parkinson and Alzheimer. So more to come later on this space. So for infectious diseases, we didn't have much information until quite recently. We know that ferrets have been trained to detect avian influenza in the US. Rats have been trained and are successfully used in Tanzania to screen people for tuberculosis. They're actually bumping the sensitivity of the suite of tests that are being used in Tanzania to detect children with tuberculosis. They are 70 more sensitive than the sputum smear microscopy. We also know that even insects can actually detect disease. During the COVID outbreak, researchers have trained bees to detect COVID, and it was also a successful outcome for this study. Now, what for COVID? At the beginning, we didn't know if COVID was actually having a special scent that could be sensed by animals. So we wanted to investigate that. We decided to use dogs. Shall we be surprised that dogs are actually good at detecting disease? No, they are all coming from wolves. And we know that wolves were actually hunting their prey using their olfactory abilities. Not only stressed animal release epinephrine that can be sensed by the predators, but they also prove that a diseased animal were releasing a suite of volatile organic compound that could be sensed by the wolves. Dogs are anatomically and physiologically equipped to detect and process scent. It's actually interesting. They breathe in stereo. So they actually breathe with both nostrils, sometimes at different time. And when the air is coming through the nostril, it's kind of divided through two different pathways. One is for going through the respiratory pathway, and the other one is going through the olfactory pathway. And you can see in this diagram, the olfactory path line in red is actually extremely vast. They've got an olfactory epithelium that is very well equipped with highly specialized olfactory receptor cells, approximately 300 million, while we only got 5 million. When all the information is captured by those olfactory cells, it then goes to the olfactory bulb, which is also much more developed than the one of a human. That's the reason why dogs have been used for a plethora of tasks, from detecting drugs, counterfeit money, electronic material at borders, but also meat or seeds or plant material. They're also used in conservation to detect or to look for endangered animals. Obviously, the rescue team use them to find people during earthquake or fires or any emergency situation. And they're also being used to detect truffle. A bit better than the pig, because pig are also used to detect truffle, but you have to be fast enough to remove the pig before it eats all the truffle. So from this literature that I read, but at that time, during the COVID, or the very beginning of the COVID pandemic, I didn't have any information about the sense of like the smell of COVID. I didn't know if animal could actually detect COVID. And before embarking in this adventure, I wanted to have a bit more certainty that it could be done. So I contacted one of my colleagues, Professor Dominique Grandjean. I translated our conversation in English because normally we speak in French together. And I asked him, did you know if dog can detect COVID-19? And do you have any information about how accurate they are? He told me, I cannot tell you how accurate they are, but I can tell you that we've tried at the very beginning of the pandemic to see if they can discriminate between positive and negative sweat sample of people affected with COVID. And after imprinting the dog with positive sample, and they detected. So they were able to detect someone that was positive. He didn't know how it was working and didn't know how good it was working, but that was, for me, an answer that was good enough to start the scientific journey of trying to train dogs in Australia. We have to know that in Australia, we didn't have COVID. So training dog for COVID-19 at the beginning was challenging, but I will go through this a bit later in a few more slides. How do we train dogs? So us, we developed an apparatus. You can see this stainless steel scent chamber. Initially, we trained the dog using something else than a medical sample. We actually use Kong, which is a plastic with a special scent. And we told the dog, when you find the target odor, so this is the target odor in this first instance, you have to sit and stare. When you find the target odor among all the apparatus, you sit and stare. And when you do that, we are nice to you and we give you a reward, which can be a toy, or it can be a piece of food. They're all Labradors, so they actually really were keen on their food. And then we imprinted the dog with sweat sample. And that's a little video. It's working. So sweat sample, we took sweat sample because it was non-infectious. We did 30 PCR on sweat sample from people that were hospitalized due to COVID, and all the PCR came back negative. So we had a bit more confidence in the fact that we were not bringing COVID to Australia. We actually imported from other countries more than 3,000 sweat sample from positive and negative patients. And we imprinted dog. So how you imprint dog? You give a sweat sample, you tell him sniff, he sniff, you will sit and you say, yes, that's the answer we want. I give you a reward. You don't want to do that with only one sample. You want to do that with a lot of samples to make sure that he's actually not learning to detect one person or one ethnic group or one sex. He has to actually come from people with disease or with symptoms and without symptoms, from as many strains as possible, as many ethnic group as possible. So that's what we did. When we, so that's the study. we actually got samples from France, from the UAE, and later on from Australia. When we did that, we actually had two groups of dogs, and we partnered with the Australian Border Force. So the Australian Border Force, in their mandate, they have to protect Australia from incursion of threats, including diseases. So they partnered, they were actually the primary partner for this entire study. We had, all dogs were donated by the Australian Border Force. We had a group of 8 dogs in Melbourne, where their headquarters is, and 7 dogs in Adelaide at Roseworthy campus. So after imprinting the dog, we actually wanted to know how accurate they were. So we did a lot of blinded tests where we put samples in all those apparatus. So the apparatus could be in line or in a circular fashion. And we asked the dog find the positive sample. There was either one positive sample or no positive sample in the apparatus. And the negative samples were coming from people who were without any symptoms and with a recent negative PCR of less than 48 hours. The positive samples were coming from people with or without positive clinical symptoms and with a recent positive PCR of a cycle threshold that was between 20, that was before, below 25. So that's how it looked like. So the handler was totally blinded. So the handler didn't know where the positive sample was, or if there was a positive sample. So that was, I'll show you the first video that was in Roseworthy. The second is actually done by ABF in Melbourne. So you see, the handler was doing a gesture, saying this is the answer I'm going to give, saying this is the positive sample. We didn't want to keep reinforcing a dog who was doing a mistake. So someone else who was not blinded will then say to the dog, yes, you are correct. So we've completed 1,333 runs with 1,200 positive cases, and all were double-blinded. The results are there. So across different dogs, we had different results. That's the reason why you've got name of the dogs and which team do they belong to. The sensitivity ranged from 87.6% to 100%. So we had one Border Force dog that was initially trained to detect explosive, that actually managed to have a sensitivity of 100%, meaning that the specificity was actually lower than the other dog. So if you had a dog very sensitive, normally the specificity was a bit below the other. And if you had a dog very specific, the sensitivity was not as great. And the specificity was ranging from 96 to 100%, also depending on the dog. That gave us enough confidence to say that the dogs could be a potent tool to be used at an airport. So we did our first trial at Sydney Airport. So the people arriving were going to go in midi-hotels anyway, and we wanted to know if the dog could detect them upon arrival. So this was actually not a true screening, because we were not allowed to truly screen people and take a video at the stage. So we were just like a training at the border, trying to distract the dog. This where everything fell down for me. And I had a wonderful study, everything was working, and we arrived at the airport, and the dog indicated on people that were PCR negative at day 1, and didn't indicate at people that were PCR positive. My whole world fell apart. It was 3 intensive weeks of me scratching my head at night and thinking, what's happening? Why are they not performing as well as they were performing in clinical settings? Then one night, I had the little bulb light bulb. I thought that maybe the dog were detecting people only when there was active replication of the virus inside the organism. The reason why it came is that we had one case that was dog positive upon arrival, PCR negative, and 3 days later the person was PCR positive. And I was like, maybe the dog is actually detecting people that are replicating, and the PCR might just be a bit too late to pick it up. And for the people that are negative, maybe they are not replicating anymore, but depending on the cycle threshold that you use, they actually are picking it up with PCR, but the dog are not picking it up anymore. And that was the case. So we could go back to the border force and very high authorities to say, no, we didn't waste all that money. Actually, it does make sense. So to actually prove that, we didn't need to know if it was PCR positive or negative. We need to know if it's PCR positive, what the cycle threshold did you use? And at what stage of the infection are we? So we did that with a sample from New South Wales. It was giving us enough confidence that yes, that was actually the key of this detection. And it allowed us to actually continue to a trial at Adelaide Airport. And when we knew that, everything was much easier, meaning that we could still, when a dog was detecting someone that was PCR negative, we were just saying, okay, the dog might be right, here an answer, and we'll decide later if the dog was right or not. So we've done that on while understanding the strengths and the limitation of the dogs, but we had two major issues at the airport. We were still relying on sweat sample. This is a very cumbersome process and actually slowing down the flow of passenger at the airport. First, we have to ask people, with this, it was still voluntary, they have to actually agree to participate. So first, do you want to enroll in this study? At the airport, only 12% of people said yes. Vast majority didn't want. They were going to go in Mediotel anyway, but they didn't want to have a positive result that might impact where they were going to go. So a few people agreed, and then when they agreed upon participating on the study, they were. It was a very slow process to actually get the dog to get the sample and to screen. When I'm saying very slow, it took 15 minutes from the sample to the results, but it was still too slow for an airport environment. The other problem is that training dogs using sweat sample is also very logistically difficult, because you need to actually get the buy-in from hospital. You need to make sure that you get your negative and your positive sample from the same spot. So it's actually a very cumbersome process too. So to alleviate this training issue, we developed a training aid. We didn't want to actually use sweat sample anymore. We wanted to use a surrogate scent of COVID-19. And we got very lucky because we tried a few things. So this was a trial and error. What are they detecting? We didn't know. We actually did a cocktail of proteins and amino acids that are coming from the virus. So it's a viral cocktail. And we imprinted initially only 2 dogs from the Australian Border Force. So we were preparing the cocktail actually with the reference lab who was culturing COVID and so on. Obviously, gamma irradiating it to make sure that it was inactivated. We imprinted 2 dogs, actually ABF imprinted 2 dogs with this cocktail. So you tell them to sniff, they sit, and you say, that's what you need to look for. And then we sent samples from New South Wales Mediotel, negative and positive sample to the border force and we didn't tell them where is the positive sample. Find the positive sample from your dog that have been only trained with this training aid. And it's actually, I think, the only day I cried of joy in this entire project. I cried a lot, but that was the only day I cried of joy, was when I received a call from Australian. They actually first didn't call me, they just sent a video. And I could see the dog, and the first dog sat where the positive sample was. That means that we managed to actually develop a surrogate scent of COVID-19 that was going to be used to train dog. The second dog did the same, and they had a few samples, and it worked on all samples. So we had a lot of confidence that we actually developed a tool that could be used to upscale the dog, to upscale the dog team, to upscale this capability. And that's going to be, published soon. The second part was we don't want to rely on sweat sample. We want people to be sniffed directly and get the results straight away. Now that we had a non-infectious training aid, we could actually put it wherever we wanted. So we started by actually putting the training aid on socks of people. So here you see they've got no shoes. And we tell the dogs, okay, find where the training aid is. And he found it, and he gets his reward, which is like playing with his handler. This worked very well. Now we need to move from the training aid to the real sample. Sorry, that's going to show you what we were hoping to do at the airport, but at that point, it was decided that everyone could come in Australia without further COVID tests, so we didn't have to do that at the airport. But we continued to work, and that's actually a true screening with people, no training aid. This is actually truly COVID. So this was my daughter. She was coming as a negative patient because I actually didn't know she was positive until we put her on this training. So we screened a lot of people. We also then went to the dormitories. In Roseworthy, we had a lot of COVID in the dormitories on campus. So what we did is we were screening the students. And, oops, sorry, I want you to see this video. No, it doesn't want to work. So again, that's a true screening. The dog is sniffing feet of everyone. So this person was then sent for a PCR, which was positive. So we actually gained a lot of confidence that the dog were able to detect people that were positive. So that's when we approached SA Pathology again. So SA Pathology has been instrumental in this project. They really supported us during the trial at the airport. And we say, I think now we are ready to try in an hospital. So we did so at the Lyle McWane Hospital at the emergency department. So the way the people were screened was a bit different. Instead of having a line of people, the dog were asked to screen one person and to give an answer, positive or negative. And then the dog was removed from the person, and then we waited for the next person to arrive. After that, the person was going for a rat test because every inpatient was going for a rat test anyway at the hospital. And some of them were going for a PCR after. We approached 740 patients during this trial. We had a very good acceptance rate of around 90%. The reason why people accepted is because when they come to an hospital, they actually want to know what is wrong. So it's not like when they arrive in an airport and they just want to get away as soon as possible. And so we had a total of 613 participants that were screened by the dog. And that's to just show you the level of agreement between the dog and the rat or PCR. Rat or PCR actually at the hospital were very similar. It seemed that the rat tests were as good, in our experiments, the rat tests were as good as a PCR to detect people with COVID-19. So dogs were ranging from 90% to 95% agreement with the tests that were done after the screening. So I think it's a little revolution because that means that we can diagnose and screen people using volatile organic compounds. And I think it can have many applications for other diseases, non-infectious like cancer and neurodegenerative disease, but also for other infectious diseases. We are currently working on Q fever, and I can tell you that dog can differentiate culture of Q fever from other bacteria, from E. coli, for instance. I think it will be useful for other diseases that can have a pandemic potential, especially now that we've got the know-how to actually develop training aids. We've got another project coming soon where we are going to see if dogs can detect people with long-term COVID, with the hypothesis that maybe long-term COVID is linked to an infection, a remnant infection somewhere in the body, likely in the gastrointestinal tract. That is obviously not picked up by nasopharyngeal PCR, but that could potentially be picked up by dogs. So more to come on this one later. The adventure of the COVID dog was obviously very exciting. Dogs is still a very good tool, and they are at this stage more sensitive than electronic nose. Having said that, in the long term, I think it will be better to have a machine because dogs require handlers, dog can get sick, and dog can get bored. So what happened at the hospital is that dogs is a very high throughput tool. And so what we can do with dog is roughly 200 to 300 people an hour. They do 10 people in 15 seconds. And that's how they work, and that's what they like. At the hospital, we were doing 50 to 60 people a day. Way too slow for dogs. And actually, that was something we had to navigate and to do something else during the experiment. We had to actually sometimes remove the dog from the hospital and go to the dormitories, where they actually were screening a lot of people in a very short period of time to make sure that they kept motivated. So in the long run, I think electronic nose should be something that we need to develop and invest much more. We are currently looking at mass spectrometer to look at profile of diseases, but that will be another talk in hopefully a year or two. So the next phase is going to be the other diseases and the long-term COVID. And my video for the acknowledgement doesn't seem to work because we had many people and industries involved, or not industries, entities. So Border Force, Australian Border Force was the first one, but we also had New South Wales Health, SA Health, SA Pathology, and we were sharing all our data in real time with a wide range of countries. So even so, as academic, we always try to publish. Our aim at this stage, we were more pro-academic. We were just trying to make sure that whatever we were getting was shared with everyone as fast as possible. And obviously, we should thank the thousands of volunteers that have been giving their sweat. Thank you very much, and I'm happy to answer any question you may have.
Lovely, thank you very much. That was fascinating and also very enjoyable to see all of the dogs as well. So thanks for that. So questions for Anne-Lise, and then people, if you're watching online and you'd like to ask a question, perhaps pop it in the chat and we'll see if we can extract it.
So that was an amazing talk. Mine's really more practical. What happens to the dogs now that COVID's not such a big issue?
Yeah, good thing. So we've got a dog two dogs that are going to work on another disease. So we are working on Q fever. We've got 2 dogs that are working on something totally different, but a bit connected, on illegal wildlife trade, because most of my research is actually based on disease detection and management at the wildlife-human interface, and illegal wildlife trade is one of the big issues. So 2 dogs are going to be deployed there. All the Australian Border Force dogs that were on loan for the project went back to Border Force. They are working again on explosives or weapon. We didn't take any dogs that were trained to detect drug because we didn't want to have any interference with people who might have drug on them and then COVID too. So yeah, and then we've got 3 dogs that were good, but we felt that they were not performing at that level and that they've been donated, and they are currently couch potatoes.
Thank you for the wonderful talk. Also a bit practical as well. How does it work if there's more than one person with COVID in the line?
Yeah, very good question. So we did have that, especially when we started to actually work in the dormitories. So the dog was praised for indicating one person that was positive, and then was brought back to the last point where he finished and was sent again. So we could have more than one person. And actually on the video from the dormitory, that's what happened. So the dog indicated one student, was praised for doing so, and then was brought back and said, now you continue the work. So yeah, that's a very good question. One thing I didn't mention in the challenges at the hospital, but now you make me think about it. So we were screening one person after one person. So we didn't have this issue of having more than one person. But we realized that we sometimes had to bring dogs in a room where the person to be screened had been there for 15 minutes, sometimes 30 minutes, even an hour. And the entire room, I think, was so strong for the dog that we had what we call adverse reactions, and the dog didn't want to get in or was indicating like way before the room. So we tried to make sure that we were in a space that was well ventilated, because otherwise we had this issue of the dog being overwhelmed by the scent.
Thank you, Anne-Lise. Amazing work. So really well done. I just wonder, you've shown us many people were involved. How many people were involved in the actual project management? So all the ethics, the governance, the finance, the coordination, all those other things behind the scene, please.
That's a difficult question, because as I explained, it was a journey. So when we were doing in New South Wales, the project in New South Wales, I had one person helping with ethics and 3 persons helping with governance in the different medi-hotel, and a few nurses that were employed for the project. When we were in Adelaide, again, I was helped with 1 person for the ethics, 1 person for governance, and same thing for the hospital. So the issue is that this project I think we were kind of walking slowly and trying to go forward, but didn't know what was going to be the end point, where we're going to be stopped. So we didn't ask like $3 million for the entire project. Each time we ask a little bit of money and a little bit of support to go a little bit further. Now that we've got this understanding of what can be done and how it's working, I will put a large NHMRC grant for other diseases, where I've got a better understanding of what's going to be the different steps, and hopefully we'll have the same person who can help with ethics, governance, and so on. Because you're right, I cannot give you a number, because very often it was people helping there and there, and we kind of had to re-explain the whole project each time we were approaching another entity.
Yes, thank you. And good to see all the volunteers that were involved as well.
Yeah.
And I just wonder, one other question, how could you maybe talk just a bit about One Welfare versus One Health? You know, do dogs, because we heard initially with COVID, the dogs could catch COVID, but the dogs get used for a lot of other things too.
Yeah. How
that ties up.
So that was the fact that dog can catch, even though they are not very susceptible. The fact that dog can catch COVID is the reason why we decided to actually use sweat sample and not mask, because sweat sample were not infectious. And when they were working at the hospital, everyone had to wear a mask before being screened. Even so, there is still a risk. We did screen the dog every 3 weeks, like PCR screened the dog every 3 weeks during the whole life of this project, and none of the dog turned positive. You're right for the One Welfare, about the well-being of the dog. I have to say that when I arrived at Border Force, I didn't know how they were training dogs and how the dogs were actually being managed. I was flabbergasted when I saw the facilities. The dogs got pools, running ground, and so on. So they actually are very well treated when they are not at work. And I have to say that they do like their work. Our dogs was a bit different. They were in kennel. We had people taking care of them and so on. But in the future, I actually don't want to keep the dogs in the kennel. I think the reason why they are so driven to work is because they sometimes spend not enough time outside. The dogs that are currently working on other projects are not kennelled, and I think they are working as well. So I think it depends a bit of the scale of your project. If you've got 100 dogs, you have to kennel them, and it's good that you need to have the money to actually provide the facility like ABF does. We are a much smaller scale, so we'll just have to do it differently. Having said that, a lot of projects are kenneling dogs, and it's working, but it's just like one perception. And so I address the disease, the welfare, and that's it. Maybe I should just add one thing, which is actually not part of the project, but really kind of touched me when it happened, and it can be another use of the dog, is one person at the emergency department was on a trolley, very unwell, an elderly woman. She wanted to be screened, so she actually signed, it was still voluntary. And the dog screened her, and she was negative, but I could see that she approached her hand to touch the dog, which was normally not allowed. But in this instance, and the dog pulled to actually get to her, and we let the dog having this interaction and the person to touch the dog, which was honestly, and you don't need like to have a PhD to see that, it was comforting for this person. So it was not the purpose of this project, and I don't know if I should mention it, but I do think that having dogs in hospital should be something that needs to be further pushed.
Thank you very much.
Yep, over here.
Thank you. That was a very fun presentation. I just had a question around, of course, during COVID, we had a lot of new strains coming out. And I'm not sure quite how it works, but would you have to ever retrain a dog to a newer strain? And does that happen across lots of viruses as well?
So excellent question. So we didn't have to retrain dogs between strains, but I can report, and that's something we discussed in the article that we are preparing, is that we were doing training aids, and the training aid that we developed was with Delta. So all the dogs were being imprinted to detect Delta, the dog that went to the hospital after. And when Omicron arrived, we actually also developed an Omicron training aid, BA1 and BA2. The first time we actually gave the Omicron strain and Omicron samples, the dog detected it. But for the first time, we had dogs that were unsure. And the way you see that the dog is unsure is that the dog is sitting, but it's not committing. And we had to actually praise them, say, no, you sat and you were right. It didn't happen for any of the other strains. They were managing to go from one strain to the other without any issues, and maybe without even noticing that there was a different strain. Between Delta and Omicron, we had this little 3-minute glitch that we had to fix because maybe something was a bit different for them. If we had a totally different strain coming, we might have to retrain the dog. That's the reason why the fact that we can have a training aid is, I think, of utmost importance.
Yeah, anyone else want to ask a question? Anyone online? We've got about 30 people online. You can ask a question in the chat. Don't be shy. All right, well, if there's no more questions, then I might ask you to join me in thanking Anne-Lise. I think it's been an absolutely fascinating presentation, so thanks so much.